Welcome to the Deep Dive. Today, we're tackling a really important topic. It's right at the cutting edge of precision medicine, medulloblastoma.
Hey, it's one of the most common malignant brain tumors in children, and sadly, one of the most devastating.
The numbers are stark, aren't they? Something like 440 new cases a year just in the U.S., and mostly kids over 65% diagnosed before they're 16.
It's a profoundly serious diagnosis, no question.
Yeah.
But what's really shifted things, based on the research we've looked at, is this scientific revolution in how we even understand it.
A revolution? How so?
Well, it comes down to realizing something fundamental over the last, say, 2 decades of intense genomic work. Medulloblastoma, it's just not one single disease.
Okay.
You see, clinically, you might have 2 tumors, one in one child, one in another. Under the microscope, what we call histology, they could look pretty much identical.
Right.
But their underlying biology, the actual molecular drivers, they can be radically different. We call this intratumoral heterogeneity.
Intratumoral heterogeneity. Okay, so if the tumors are biologically different, even if they look the same, then that old one-size-fits-all approach, standard chemo, standard radiation, it's not going to cut it anymore, is it?
Exactly. That's the core insight. So our mission today really is to give you a kind of shortcut to understanding this new molecular map.
Okay, break it down for us.
We're going to unpack the 4 main molecular subgroups that everyone agrees on now: WNT, SHH, Group 3, and Group 4. And we'll talk about the omic technologies that help define them: genomics, epigenomics.
All the high-tech stuff, and crucially, how knowing this stuff actually changes things for patients, like clinical trials, risk, even radiation doses.
Absolutely. It's moving from the lab right into the clinic, changing how treatment decisions are made globally.
All right, let's dig into that molecular foundation. You mentioned histology, looking at cells under a microscope. That used to be the only way. Classic, desmoplastic, large cell anaplastic.
Yeah, that was the standard for a long time. But around the early 2000s, things started shifting towards molecular classification. Basically classifying based on specific molecular markers, not just appearance.
So what tools made that possible? I hear a lot about next-generation sequencing, NGS.
NGS is definitely key. It lets you read, tons and tons of short DNA sequences all at once. Fantastic for spotting specific gene mutations, those little spelling errors in the DNA code.
But you mentioned it's not the only tool, or maybe not the main one for routine classification.
Well, for actually getting the subgroup sorted out reliably in clinics around the world, another technology kind of emerged as the workhorse, the platform of choice. And that's DNA methylation arrays.
Okay, hang on. That seems a bit counterintuitive. If NGS reads the actual DNA code, why would arrays looking at methylation, those chemical tags on the DNA, be the standard?
It's a really good question. It boils down to 2 main things that are super important when you're trying to get labs everywhere on the same page: stability and consistency.
Stability and consistency.
Right. DNA itself is incredibly stable. You can often get good data even from tumor samples stored for years, decades sometimes. Much more stable than RNA, for example.
Okay, that makes sense for old samples.
And the consistency part is crucial. When different labs use these standardized methylation arrays to map out that epigenetic landscape, the pattern of these tags across the genome, the results are remarkably consistent. Very low technical variation between labs.
So everyone gets basically the same map, regardless of where they run the test. Standardization.
Exactly. That reliability is what allowed researchers worldwide to collaborate and build this robust consensus map of the subgroups in the first place. It really solidified around 2012.
Got it. So reliable tools led to a solid consensus. Let's list those 4 main subgroups again with their rough prevalence.
Sure. You've got WNT, the smallest group, maybe 10% of cases. Then Sonic Hedgehog, SHH, that's about 30%. Group 3 is around 25%, and the largest is Group 4, about 35%.
And historically, how did this start? You mentioned the hedgehog pathway earlier.
Yeah, that was one of the first big clues, actually. Early gene expression studies, back around 2002, started separating MB from other similar-looking childhood brain tumors. And they noticed that these desmoplastic tumors, one of the histological types, they consistently had this massive activation, this upregulation of the hedgehog signaling pathway.
Yeah.
That was really the first major molecular distinction identified, the first crack in that old idea of medulloblastoma being just one thing.
Fascinating. So right away, these molecular markers were telling you things about prognosis and biology that just looking at the cells couldn't. Let's jump into the first 2 groups then, WNT and SHH. Okay, WNT medulloblastoma, you said this one is kind of the the good news story in all this.
Absolutely. If there's a poster child for how precision diagnosis leads directly to better safer treatment, it's WNT. The big clinical takeaway is just outstanding prognosis.
How good are we talking?
We're talking 5-year survival rates of 95% or even better. It's remarkable.
Wow. And who gets these WNT tumors typically?
Demographically, it tends to be older children, adolescents, young adults. The median age is around 11, and importantly, they're much less likely to have spread to be metastatic at diagnosis.
So great outcome, less likely to spread. What's the molecular signature? What makes them WNT?
The defining genetic event in about 85% of these cases is a specific mutation, a somatic mutation, meaning it's just in the tumor in a gene called CTNB1.
CTNNB1. That encodes beta-catenin, right? Part of the WNT signaling pathway.
Exactly. That mutation hyperactivates the WNT pathway. Another common feature you see is the loss of one copy of chromosome 6. We call that monosomy 6.
So a specific gene mutation, a specific chromosome loss. And because this group is so uniform molecularly and does so well,
it allows for treatment de-escalation.
Right.
We can safely reduce the intensity of therapy, especially radiation, minimizing those long-term side effects for survivors.
Yeah, that 95% survival is amazing, but reducing the lifelong neurological damage, that's arguably just as huge for these kids.
It really is.
Okay, so that's WNT clear mutation, great prognosis, locked treatment. Now let's contrast that with the SHH subgroup. It sounds like it's more complicated.
SHH is definitely a different beast. It's defined by that activated hedgehog pathway we mentioned earlier. But the patient demographics are really interesting. It has this distinct bimodal age distribution.
Bimodal? Meaning?
Meaning it peaks in 2 different age groups.
Yeah.
Infants, so kids under 3 years old, and adults, typically over 17. It's much less common in between those ages. It's also more common in males, about a 2-to-1 ratio.
Infants and adults. That's unusual. And it's driven by mutations activating that SHH pathway.
Correct. You look for specific genetic hits that turn the pathway on. Often it's mutations that switch off the brakes genes like PTCH1 or SUFU, or mutations that jam the accelerator on like in the SMO gene.
PTCH1, SUFU, SMO. The key players in the hedgehog pathway.
Right, but here's where the complexity really impacts treatment. The patient's age and exactly which gene is mutated matters a lot.
How so? You mentioned adult SHH tumors are different.
Yeah, genetically they are distinct. Adult SHH tumors tend to have way more mutations overall, a higher mutational burden. And crucially, they very often have mutations in the TERT promoter.
TERT, that's linked to telomerase, right? Maintaining the ends of chromosomes.
Exactly. A TERT promoter mutation basically helps make the tumor cells immortal. It's a key feature that really defines most adult SHH cases and separates them biologically from the infant cases.
Okay, so if I'm a doctor with an SHH patient, I need to know more than just it's SHH. I need the age, and I need the specific gene alteration, because that affects treatment.
Precisely. Especially when considering targeted therapies like SMO inhibitors.
Ah, right. Drugs that block SMO.
Yeah. Think about it. If the mutation driving the tumor is downstream of SMO in the pathway, then blocking SMO upstream isn't going to do anything.
Makes sense. The signal's already past that point.
Exactly. So if the mutation is in PTCH1 or SMO itself, maybe a SMO inhibitor could work. But if it's further down, like in SUF, you need a totally different strategy. This molecular subtyping isn't just academic. It directly predicts whether a specific targeted drug might even have a chance of working.
Okay, so WNT and SHH seem relatively well-defined, even with SHH's complexities. But groups 3 and 4, you said they represent the biggest challenges, most common, most varied.
That's right. They were often historically just lumped together as non-WNT, non-SHH because they were harder to tease apart. Group 3 is generally considered the most aggressive.
Who gets group 3?
Primarily infants and young children, very rare in adults. It's got a strong male bias, even more than SHH. And tragically, a high percentage, maybe 30–40%, already have metastatic spread when they're diagnosed.
That's tough. Is there a key genetic hallmark for group 3?
One of the most notorious is MYC amplification. MYC is a powerful oncogene, and having extra copies of it is strongly linked to very poor outcomes in group 3. Histologically, it's also more likely to have that aggressive large cell anaplastic appearance.
Okay, MYC amplification is bad news in group 3. What about group 4, the largest group overall?
Group 4 is also common, affects all ages from kids to adults, shares that male bias, and unfortunately also has that high rate of metastasis, similar to group 3.
So they share some grim clinical features. Is there a genetic link too?
There is a very important one, a major structural chromosomal abnormality called isochromosome 17q, or I17q, It's found in over half of both group 3 and group 4 tumors.
Isochromosome 17q. What does that actually mean structurally, and what does it do?
Okay, so an isochromosome basically means you lose one entire arm of a chromosome, in this case, the short arm of 17p, and you gain a duplicated copy of the other arm, the long arm, 17q.
So you have 2 copies of the long arm, no copies of the short arm.
Exactly. Functionally, this huge structural change almost always results in loss of heterozygosity across that whole arm. You lose any diversity you had. And critically, the 17p arm that's lost contains important tumor suppressor genes, like TP53.
So I17q is a mechanism for losing key cancer protection genes, and it's common to both group 3 and group 4.
Correct. It's a major shared feature linking these two often aggressive groups.
It really sounds like the deeper researchers dig, the more complex it gets. We started with 4 groups, but now I read about 8 distinct molecular subtypes within just groups 3 and 4.
Yes. The resolution keeps increasing. For example, you mentioned subtype 8 within group 4. It's actually the most common single subtype overall. And while it often has a relatively good initial survival,
There's a but?
there's a but. It has this unique and worrying tendency for late relapse and death, years after treatment seems successful.
Wow. That requires incredibly long follow-up and vigilance.
Absolutely. And understanding why groups 3 and 4 behave the way they do, including these late relapses, means looking beyond just those classic gene mutations. We need to talk about structural variants, SVs, those big rearrangements like I17q and a fascinating mechanism called enhancer hijacking.
Enhancer hijacking. Okay, break that down for us. Sounds intriguing.
Think of enhancers like remote controls for genes. They're often located physically far away from the gene they regulate on the DNA strand. But when they get activated, they loop over and tell that specific gene to turn on, often very strongly.
Okay, a remote control for gene volume.
Kind of, yeah. Now, in enhancer hijacking, a structural variant, maybe a deletion, an inversion, a translocation, basically rewires the genome. It physically breaks the DNA and reconnects it incorrectly.
And this connects the enhancer to the wrong gene.
Precisely. It takes a powerful enhancer that normally controls some other gene, maybe one involved in development, and sticks it right next to the promoter, the on-switch region of a gene that should be silent or expressed at very low levels in that cell type. Often it's an oncogene.
So in group 3 or 4, you might hijack an enhancer and point it at genes like GFI1 or PRDM6.
Exactly. Those genes, GFI1, GFI1b, PRDM6, are common targets. The enhancer, now misplaced, drives massive overexpression of that oncogene. The gene's own code, its protein sequence, might be perfectly normal.
But it's just screamingly loud because it stole the wrong volume control.
That's a great analogy. And this isn't a rare thing. This mechanism explains something like 12 to 15% of group 3 and group 4 tumors. It really highlights that the problem in these groups often isn't a broken protein, but fundamentally messed up gene regulation.
This genomic complexity, the structural variants, the hijacking,
Yeah.
it seems like just sequencing the bulk tumor might miss crucial details. You mentioned intratumoral heterogeneity earlier, the idea that tumors are actually a mix of different cell types.
Yeah, that's a critical concept.
Huh.
Tumors aren't just monolithic blocks of cancer cells. They're complex ecosystems with different malignant cell populations, plus non-malignant cells like immune cells, stromal cells. Bulk sequencing averages everything out.
So how do you see inside that mix?
That's where techniques like single-cell RNA sequencing, scRNA-seq, have been revolutionary. Instead of sequencing the average RNA expression of the whole tumor chunk, you sequence the RNA from thousands of individual cells within it.
And what does that tell you?
It lets you map the different cell populations and even trace their developmental origins. Where did these cancer cells come from in the developing cerebellum?
Okay, so do different MB subgroups seem to arise from different cerebellar cells?
It appears so. The single-cell data suggests SHH tumors, for example, closely resemble developing granule neuron precursors, or GNPs. They seem stuck in that proliferative state.
And what about the really aggressive group 3 tumors, the MYC amplified ones?
Those seem to be composed almost entirely of very immature, undifferentiated, progenitor-like cells. They basically have zero capacity to mature or specialize. They're just stuck in grow mode.
Which explains why they're so aggressive. Locked in a primitive state. What about group 4?
Group 4 is interesting. Prototypic group 4 tumors tend to resemble more differentiated neuronal cell types like unipolar brush cells, UBCs, or certain deep cerebellar nuclei cells.
So there's a spectrum. SHH, like early precursors. Group 3, totally undifferentiated. Group 4, more differentiated, but still cancerous.
Exactly. It looks like a developmental continuum, or perhaps different points where development gets blocked or hijacked. Group 3 seems blocked very early, while Group 4 cells have progressed further before going wrong.
Okay, single-cell RNA gives us the developmental picture, but what about the proteins, the actual machinery doing the work? You mentioned proteomics. Why add that layer?
Because genes and RNA are just the blueprint and the message. Proteins are the functional units. Sometimes the amount of RNA doesn't perfectly predict the amount or activity of the protein. There are lots of controls after the RNA is made, what we call post-transcriptional mechanisms.
Explain that simply. Post-transcriptional.
It means after transcription. So the DNA gets transcribed into RNA perfectly fine, but then maybe that RNA doesn't get translated into protein very efficiently. Or maybe the protein is made, but it needs an extra modification, like a chemical tag called phosphorylation to switch it on or off. Genomics alone won't tell you about that final activity state.
So proteomics, using things like mass spectrometry, measures the actual proteins and their modifications.
Exactly. And when applied to MAB, proteomics confirmed the main subgroups, but it also revealed some finer substructures, like different flavors within SHH or Group 3, that weren't as obvious just looking at the RNA. It adds another layer of functional information.
Did it uncover any potential new targets, particularly in those tricky Group 3 or 4 tumors?
Yes, actually. A key finding in Group 4 proteomics was evidence of aberrant RTK signaling. That's receptor tyrosine kinase signaling, specifically activation of pathways involving ERBB4 and SRC.
And RTKs are often targets for cancer drugs, right?
They are. This finding was then functionally validated in mouse models, showing it was really driving tumor growth. So it immediately pointed towards a potential therapeutic strategy for Group 4 using RTK inhibitors.
Okay, this is a huge amount of molecular details, subgroups, subtypes, pathways, hijacking, single cells, proteins. How does this all translate into actual changes in the clinic? You mentioned trials like SJMB12 are incorporating this.
Absolutely. This isn't just research anymore. It's directly informing treatment protocols. The biggest impact so far is definitely in risk-adapted therapy.
Meaning tailoring treatment intensity based on the molecular risk.
Precisely. Take the WNT subgroup. We know they have that excellent prognosis. So in trials like SJMB12 and others globally, patients confirmed to have low-risk WNT tumors receive significantly reduced craniospinal axis irradiation, CSI.
Remind us what CSI is and what reduced means here.
CSI is radiation targeting the entire brain and spinal cord because MB cells can spread through the spinal fluid. Standard dose might be 23.4 GAG. For low-risk WNT, protocols are now using doses as low as 15 to 18 JAG.
That's a big reduction. And gray is the unit of absorbed radiation dose. Less radiation, especially to a developing brain and spine, that means fewer long-term side effects.
Exactly. Less impact on cognition, hormones, hearing, and lower risk of secondary cancers later in life. It's a massive benefit derived directly from knowing the molecular subgroup. That's precision medicine in action.
Okay, huge win for WNT patients. What about SHH? You mentioned targeted SMO inhibitors like vismodegib.
Right. For SHH, the precision is about who gets the targeted drug. Vismodegib can be effective, but only if the mutation is upstream in the pathway, like in PDCH1 or SMO. And crucially, there's a major safety concern.
Which is?
These drugs interfere with the normal hedgehog pathway, which is vital for bone development. So giving SMO inhibitors to young children whose bones are still growing can cause their growth plates to fuse permanently.
Oh, wow. So it can stunt their growth irreversibly.
It's serious permanent side effect.
Yeah.
Because of that risk, these drugs are generally reserved for skeletally mature patients, adolescents, or adults whose growth plates have already closed or used very carefully in specific high-risk situations in younger kids where the tumor risk outweighs the growth risk.
So again, molecular details, subgroup, and specific mutation and patient age are critical for deciding treatment. It's incredibly precise.
It has to be. And for groups 3 and 4, while we don't have targeted therapies as established as SMO inhibitors yet, identifying things like GFI1 activation via enhancer hijacking, that points toward potential future drugs, maybe like LSD1 inhibitors, which could target that pathway, or the RTK inhibitors for group 4 we discussed. It gives us a roadmap for developing smarter, hopefully less toxic, trials for these tougher groups.
And finally, one last critical piece for families facing this diagnosis: hereditary risk. This isn't always just bad luck, right?
No, absolutely not. This is super important. We now know that a significant portion of MB cases are linked to inherited germline mutations. The rates are especially high in SHHMB, maybe 25% or even higher.
25%. Linked to what conditions?
Often things like Gorlin syndrome, caused by germline PTCH1 mutations, or specific SUFU mutations often found in infants with SHH tumors. Even WNT-MB has a hereditary link in about 5–10% of cases, typically involving the APC gene linked to familial polyposis.
So finding MB in a child means genetic testing, and counseling isn't just optional, it's essential.
It's mandatory. Finding a germline predisposition has huge implications not just for the patient's treatment and surveillance, but for their parents, siblings, and the wider family. It requires lifelong monitoring.
Wow. We've really covered a lot of ground. From just 4 broad histological types to now at least 8 molecular subtypes, digging into structural variants, enhancer hijacking, single-cell origins, proteomics, it sounds incredibly complex.
It is complex, no doubt, but that complexity, that deep understanding, is precisely what allows us to move forward. It's the necessary path towards reducing the devastating toxicity for lower-risk kids and developing truly effective targeted therapies for the high-risk groups.
So the goal isn't just identifying risk, but acting on it intelligently.
Exactly. And the field isn't stopping. We're moving rapidly beyond just profiling the bulk tumor towards routine single-cell analysis. Why? Because we know that relapse, metastasis, treatment failure, those are often driven by tiny populations of resistant cells.
The rare clones that survive therapy and come back later.
Precisely. Understanding those rare cells, maybe the ones stuck in that undifferentiated state we talk about, or ones that acquire new mutations, that's the next frontier to truly conquer this disease and push towards that 100% cure rate.
Which brings us back to that challenging group 4. It's the most common, often has those complex structural changes like enhancer hijacking rather than simple mutations, and carries that ominous risk of late relapse. Sneaky tumors, as you said.
They are. The initial response might look good, but something is lurking.
So that leads us to our final thought, a question for you, the listener, to ponder. Given everything we've discussed about Group 4, its prevalence, its reliance on mechanisms like enhancer hijacking, and its unique pattern of late failure, what hidden molecular time bombs, what subtle cellular states or vulnerabilities might still be hiding within these complex tumors that only that deeper single-cell functional profiling can ultimately uncover to finally get ahead of relapse and cure every child?